Hello, my name is Daniel Tsai. I am a senior at Duke University, where I am a member of the Keyhart Laboratory, and today I will be presenting my project titled Investigating D. melanogaster cell sheet morphogenesis, a live imaging study of two genomic regions of the 2L chromosome important for dorsal closure. I've been working on this project for over 2 years now with Stephanie Fogerson, Janice Crawford, and Dr. Daniel Keyhart. Before we jump into the project itself, let me explain a little about the relationship between D. melanogaster and cell sheet morphogenesis. The field of cell sheet morphogenesis investigates how sheets of cells, opposed to individual cells, develop their shape and function. Dysfunction in humans has been linked to many systemic congenital or developmental conditions. D. melanogaster has proven to be an effective model to study cell sheet morphogenesis because it is capable of being genetically manipulated, amenable to live imaging, and has a relatively short generation time. More importantly, despite gross morphological differences, D. melanogaster shares many processes at the molecular, cellular, and tissue level with humans. About 65% of human disease-causing genes have a functional homologue in D. melanogaster, with a majority being expressed in an analogous tissue. Therefore, many non-functional fly genes can be replaced and rescued by human genes and vice versa. In this project, I use dorsal closure as a way to study cell sheet morphogenesis in D. melanogaster. Dorsal closure is a fundamental 3 to 4 hour process in embryogenesis. It involves 2 sheets of lateral epidermal cells zipping an elliptical hole filled with amnioserosa cells on both ends along the dorsal midline, with the greatest movement occurring at the purse string and dorsalmost lateral epidermal cells. As of 2018, there were over 140 known dorsal closure genes, which is defined as any gene that causes an abnormal phenotype when mutated. To study dorsal closure, I used a Bloomington D. melanogaster deficiency kit, which is essentially a collection of genetically modified flies. To be more specific, a deficiency is a mutation in D. melanogaster that removes long stretches of continuous DNA on a chromosome. The kit consists of 473 deficiencies, or genetically modified flies, that collectively delete 98.4% of annotated genes in the D. melanogaster genome. The kit is subdivided into 6 groups, each covering a different arm of an autosomal chromosome, or the X chromosome. The diagram below, pulled from Bloomington's website, is an example of a deficiency map. The picture itself represents the arm of a chromosome. The red bar at the bottom is the part of the chromosome that was deleted by a deficiency, and the arrows at the top are genes those sections of DNA encode. A recent screen of the left arm of the second chromosome successfully bred viable homozygotic deficient embryos for 99 out of 108 available deficiencies. A homozygous deficient embryo implies the embryo is lacking the deficient region on both copies of the given chromosome. Studying these embryos is valuable because they illustrate what happens when an embryo has no working copies of a given gene. By crossing and imaging 99 out of 108 available deficiencies, this screen examined 87.2% of the 98.9% of genes affected by the 2L kit. It discovered 49 deficiencies that produced significant DC defects when homozygous in a Ubi-cadherin-GFP background, and it discovered 27 novel DC genes. This project further investigates 2 of the 27 deficiencies in the 2L screen that did not contain a known DC gene using a similar method of confocal microscopy imaging. It's worth mentioning I only outlined 1 deficiency in my abstract, but will be presenting a second one today. The first is called DF2L27, and the second is called DF2L38. In accordance with the 2L deficiency screen, these deficiencies, which we will now call parent deficiencies, are numbered based on the chromosomal position of their breakpoints, with lower numbers indicating closer proximity to the telomere. These 2 parent deficiencies were selected for 2 reasons. First, their embryos displayed a severe DC phenotype when homozygous in a Ubi-cadherin-GFP background. Second, their genomic regions are saturated with overlapping subdeficiencies. Subdeficiencies, which you can see in the same example map below, are just deficiencies that partially overlap the parent. So, if you crossed flies of a parent and subdeficiency, you would get progeny that is homozygous deficient for some genes and heterozygous deficient for the rest of the genes of the parent deficiency. Comparing dorsal closure in these transheterozygous embryos to the homozygous parent is valuable because if they have the same phenotype, we know it is a result of genes in the homozygous deficient region. Performing these crosses leads to my ultimate goal of identifying the novel DC genes in each parent deficiency responsible for their respective DC phenotype. This is a diagram of the crossing scheme we use to acquire the transheterozygous embryos. The purpose of the first cross is to balance the parent deficiency and subdeficiency over a marked balancer using an imaging line of Ubi-cadherin-GFP on the 3rd chromosome. Males of the parent deficiency and subdeficiency are crossed with females of an imaging line on standard cornmeal and molasses fly food at 25 degrees Celsius for 10 to 14 days. The progeny are selected by picking flies with traits correlating with TGC, like curly wings, while selecting away from flies with traits correlating with SCO, like missing scutellar bristles. The second cross combines 15 to 20 virgin females of the first parent deficiency cross and 7 to 10 males of the first subdeficiency cross in a cylindrical cage with a removable juice agar plate at 25 degrees Celsius for 48 to 72 hours. This results in embryos that are transheterozygous for the loci being tested. If the progeny of the second cross phenocopies or exhibits the same DC phenotype as a homozygous parent, it can be assumed the genes causing the dorsal closure phenotype are within the overlapping or shared region of the cross between the parent and subdeficiency. The crosses undergo a timed collection where embryos laid on the juice agar plates are collected at 25 degrees Celsius over 4 hours and then aged at 16 degrees Celsius for 24 hours until embryos are in late germ band retraction. Embryos are washed and decoordinated with bleach, then manually sorted under a dissecting microscope equipped to detect green fluorescence, selecting for embryos in early DC and away from the fluorescence of the marked balancer. Wild-type embryos crossed to an imaging line are also collected as controls. Embryos are mounted on an inverted confocal microscope equipped to capture 4D imaging. Then, over 120 time-lapse images of each embryo are captured over a 4-hour period until DC completes or has clearly failed. Z stacks are compiled into maximum intensity projection movies using Metamorph and Fiji software, and then scored for defects in morphological tissue movements. The results of this project are quite exciting. First, let's talk about parent deficiency 27. 6 homozygous embryos of this deficiency were imaged during the 2L screen, and the most notable defects were the presence of cuboidal LE cells, development of a cigar-shaped dorsal opening, and AS cells falling apart. For this project, DF27 was crossed with DF27A, which, as you can see in the diagram, creates progeny with a 54 kilobase pair homozygous deficient region with 8 genes and a 64 kilobase pair heterozygous deficient region with 11 genes. 15 transheterozygous embryos were imaged from this cross. As you can see from the table, the transheterozygous embryos appear to phenocopy the cuboidal LE cell and cigar-shaped dorsal opening defects at a high penetrance, but not the AS cells falling apart. On the right, I show an example of the progression of an embryo from each cross. So, from these results, we can conclude this cross displays a partial phenocopy for the cuboidal LE cell and cigar-shaped dorsal opening defects. Therefore, the genes responsible for those defects are located within the 8-gene homozygous deficient region. 2 candidate genes in this region I've hypothesized based on a literature review are BOL and BARC. BOL is especially intriguing based on its high expression in dorsal closure and its intrinsic relationship with the known DC gene wingless. Correspondingly, the genes responsible for the increased severity, which is particularly evident in the lack of AS cells falling apart, are located within the 11-gene heterozygous deficient region. 3 candidate genes I've hypothesized are sloppy pair 1 and 2 and CAPU. The 2L screen already hypothesized the loss of sloppy pair genes contributes to the reduced elongation that leads to cuboidal LE cells. Now, let's talk about parent deficiency 38. 7 homozygous embryos of this deficiency were imaged during the 2L screen, and the most notable defects were the presence of a jagged leading edge, abnormally shaped AS cells, and AS cells falling apart. For this project, we used the same logic as before to analyze crosses between DF38 and DF38A, B, C, and D. The cross between 38 and 38A produced 7 embryos and a partial phenocopy for the jagged leading edge defect. 38 and 38B produced 6 embryos and a partial phenocopy for the abnormally shaped AS cell defect. 38 and 38C produced 8 embryos and a partial phenocopy for both the jagged leading edge and abnormally shaped AS cell defects. Lastly, 38 and 38D produced 7 embryos and a partial phenocopy for the abnormally shaped AS cell defect. On the next slide, I show an example of the progression of an embryo from each cross. Because of the time restriction, I'm going to move on from the slide, but I encourage everyone to take a look at it after. 3 major conclusions can be drawn from these results. First, genes for the abnormal AS cell-shaped defect are located in the 8-gene overlap between 38 and 38B, C, D. 1 candidate gene I've hypothesized for that region is LAM. Second, genes for the jagged leading edge defect are located in the 2-gene overlap between DF38 and 38A, and 4-gene overlap between DF38 and 38C, but not B or D. 2 candidate genes I've hypothesized for that region is ocelin and CG14024. Lastly, severity in the parent phenotype is a result of a combination of multiple genes located throughout 38. 3 candidate genes I've hypothesized for this are fusyl, CYP, and MID. I encourage anyone who is interested to read the descriptions of these genes on the slide. Moving forward, alternative crosses with mutant allele and rescue constructs will be used to study the role of individual candidate DC genes. I want to thank everyone in the Keyhart Lab back at Duke, JYI for letting me present, and of course all the flies who dedicated their lives for this project. And with that, I thank you for listening. I hope you enjoyed learning about fruit flies as much as I have.